Thank you for inviting me to participate on this meeting today. I'm going to be talking to you about the carcinomas of unknown primary or CUP consortium that we have set up at NCI with the help of Manel Esteller, who's going to be talking after me, I think. I probably don't need to tell this audience, but I'll give a little background on what carcinomas of unknown primary are. Basically between 3 and 10%, and lately has been about 3% of all worldwide diagnosed cancers, are presenting as metastatic and distant tumors for which the primary malignancy is not found. The metastases they clinically manifest without the primary tumor, which is uncommon. Normally one finds the primaries and then the metastases. And the probably reasons for these unknown primaries are either the primary cancer is very small and it grows slowly and it cannot be identified or has not been identified. The body immune system has taken care of the primary, which is not unlikely. The primary cancer was removed during surgery for another condition, and then it was not identified when it was removed, and thus it's no longer present, and we cannot tell what the primary was. Clinically, carcinomas of unknown primary are defined as a histologically confirmed malignant tissue that is incompatible with the primary neoplasm in the biopsy area. Basically, it's something that does not look like the surrounding tissue, so it cannot have come from the tissue where it's found. So normally, the investigation of a possibility of a tumor follows the report of symptoms that the patient may have, such as a lump or a thickening in any part of the body. That's normally when carcinomas of unknown primaries are found in lymph nodes. Pain that is in one part of the body and does not go away. A cough, it could be, if it's found in the lungs. Change in bowel or bladder habits, that is in the rare cases in which it can be found in colon or in the bladder. Unusual bleeding. So all these things that are not explained, all the symptoms that are not explained when investigated, sometimes yield the discovery of carcinoma for which the primary is not known. The site of origin of the tumor has been easier to determine in recent years because the cells seem to retain a cell of origin epigenetic mark, kind of a signature that reflects where these cells came from. And it normally has been found through methylation analysis because the methylation marks of the cells persist even after malignant transformation. So studies performing in cancers of unknown primaries have preliminarily shown that administration of site-specific treatment when the original site of the tumor can be identified by these methylation or even expression studies yield a lot better result, a lot better prognostic for the patient than if just generic therapy is given to them. So it is important to try to figure out where these tumors are coming from. Manel is going to probably be talking about this. I imagine he will, which is his work showing that the epigenetic profile allows to identify in the vast majority of CUPs what is the cell of origin. And so the problem about identifying the cell of origin in order to have better treatment kind of has been solved. And so the question will be, why are we doing this? The proposal that we have and basically has been approved and funded for a CUP consortium, the idea is to study carcinomas of unknown primaries fully by looking at copy number variations, DNA methylation, RNA sequencing, DNA sequencing, and analyze about 500 cases. of clinically well-characterized CAPs to improve this unmet need not only of therapy, but also to understand why do these tumors appear, and they behave quite different from others. Initially, the extensive characterization of the cases that were already accrued by Manel and his group and his consortium are going to be characterized in the cases that they have enough tissue for that, but we are also going to be characterizing additional cases that will be collected by the clinical members of the consortium on a prospective basis. The methodology of the consortium is basically, as I said, cases will come some from retrospective collection, some from prospective collection. All cases, every single one of them will have extensive clinical information, treatment, relapses, outcome that will allow to stratify the cases when we do the analysis of the molecular information that we are generating to investigate, the underpinnings of these tumors. And all cases will be characterized by exome, total RNA, and whole genome sequencing, as well as DNA methylation, using the DNA pipeline, the NCI pipeline. And all data will be deposited in the NCI Genomics Data Commons. This pipeline that I alluded to is shown in this slide. Basically, you see up there at the top, you have the hospitals, basically, where the patients are seen, the samples are collected, the clinical data are collected, and the samples and the clinical data are transferred to two entities that fall under the Biospecimen Core Resource, the BCR. One is the Biospecimen Processing Center that takes the samples and creates the DNA and the RNA needed for characterization. And the other is the Clinical Data Center, the CDC, that takes the clinical data, cleans them up, consults with the primary sites to see if things are found to be inconsistent, and cleans out the data, and the data then it gets sent directly to the Genomics Data Commons, which is represented here by that stack of computer disks. The analytes that are created by the BPC go to our sequencing centers. Right now, we have 3 different sequencing centers, 4 really, that will create exome, whole genome, and methylation from the DNA side, and RNA sequencing, total RNA sequencing from the RNA side, as well as microRNA. And so once all that data is created for every single sample, the data is submitted to the GDC, to the Genomics Data Commons. The Genomics Data Commons will align all the data to the genome and will call the data, finding the mutations, finding the levels of expression, all the things that need to be done before that data is exposed to a group of highly trained molecular specialists that we group under the name of the Genomic Data Analysis Network. These are people that are really the world experts on each one of the modalities, and they do the analysis of copy number changes, expression, mutation, signatures, whatever really you need, they can provide. And they, along with the clinical participants that have been providing the samples, and they are the disease experts, form what we call the analysis working group. And this analysis working group basically guides the analysis and produces the analysis of this data that will end up resulting in a manuscript that describes the resource. All of that data goes back to the Genomic Data Commons, and then the Genomic Data Commons exposes to the research community at large that can use the data to whatever they want without any permissions other than requiring permission to access when the data that they request is control access data, and the only control access data that we have is raw sequencing. The reasons raw sequences are consider control is because you could identify the potential donors through the use of that data. So you have to promise basically that you're not going to try to do that and that you are not going to distribute the data any further. Once you did that, you get the access to the data, and basically you can see the entire data set. In fact, we create so much derived data that is exposed at the Genomic Data Commons that most researchers in the world actually don't need to ask for control access, because the only thing that is under control access is raw data, raw sequencing data. And trust me, you don't want to do this alignment again. We have done it in the standard ways, and we do run a lot of different pipelines to do the call. So it's very, very rare that anybody actually needs the raw sequencing in order to use our data. So why are we doing this? It's because we have some hypotheses and some questions that we would like to answer. The whole idea of the project is to answer a bunch of different questions that are related directly to CAPS, but also related to the processes of metastasis, which are really, we all agree, metastases are what kill the patients, and we know very little about how metastases happen. And so this is an opportunity to see if there's any common biological processes that play in these tumors and that differ from their origins, from cell origin, and they have obviously a different biology, but then they must have something in common that allows them to behave in the way they do. What genes are at play in these cases, and what are the different from the standard tumors that have the same origin, and so that will give us an idea. Do these tumors display different immunogenic profile? What can we learn about the metastatic process in general? And so I think if we do get enough samples to do this project, we are going to help not just the CAP community, but we're going to help the cancer biology community at large by getting to know processes in tumors that we know very little of. With that, I would like to take any questions if you have them, and I thank you. I thank the organizers for inviting me and to talk to you. Thanks.